3 teams are ready to go. So jury, do you have your notebooks ready? And presenters, good luck. First up, we have Seekyo Therapeutics. They developed the next generation of chemotherapies. By targeting the microenvironment of solid tumors while sparing healthy tissue, they provide an efficient treatment without any adverse effect, improving the quality of life for so many cancer patients. Joining us today is Uri Shedbun. Good luck. There you go.
Good afternoon. To your opinion, what is the biggest fear of patients with cancer? It's chemotherapy. Chemotherapy treatment itself. Will it hurt? Will I lose my hair? Good afternoon. I'm Uri Shedbun, CEO of Seekyo. And at Seekyo, we developed a chemotherapy which is highly selective against tumor and safe for patients. Every day, 5,000 people will be diagnosed with cancer. Half of them will undergo chemotherapy. And for most of them, that would be the best therapeutic option. But the truth is that standard chemotherapy is a very aggressive treatment. So how can we make chemotherapy safe and efficient at the same time? Selectivity is key. Scientists have been working on that for the past 20 years with limited success. At Seekyo, we've developed a new approach which enables to specifically target the tumor and be safe at the same time. We call it smart chemotherapy. Let me show you how it works. We invented a molecular platform on which we link 3 cytotoxic elements. The platform then will be driven in the blood thanks to the liaison to the albumin. Albumin is a well-known and characterized drug carrier. So Sky1, that's his name, has this molecular structure. But let me show you how it works in real life. Sky1 is injected into the blood, links to the albumin, and then will be driven into the bloodstream down to the tumor. Around the tumor and induced by the tumor itself, there are modifications of the blood vessel endothelium, which allows Sky1 to get inside and accumulate only there. At the same time, the cytotoxic agent will be released thanks to an enzyme called beta-glucuronidase, which also is only expressed at the tumor microenvironment, only in solid tumor. So once the cytotoxic agent is released, it gets inside the cancer cells and destroys them, and only then. So we know he works. We've tested that on animals. We've tested that on pancreas models. Pancreas is a very difficult cancer to treat. So we are able to reduce the size of the tumor. It's safe. It's well tolerated. We also compared ourselves to the competition, to existing solution, and a specific class of drugs called the ADC, antibody drugs conjugate. And today, we are better than anything else out there. That gives us a unique position into the marketplace. In order to maintain that unique position, we need also a strong IP. That's what also we have developed. Strong IP position and strategy in licensing, filing new patent, and also considering new patents such as diagnostic tests. So let's talk a bit about the business, the market. Today, the market, the patient are waiting for a selective chemotherapy able to target solid cancer. It's exactly what Seekyo does by targeting the pancreas cancer, the triple negative, the lung, and the colorectal. It's a very active market of 15 billion by 2030, with an annual growth of 30%. We closely worked with our ecosystem, and last year we raised 800,000 euros. Today, I'm here to accelerate the clinical development. I'm here to raise 15 million in order to bring Sky1 up to the end of phase 2, which is the inflection point, which is the time on which we will license the art out and make return to investments. So we need a good science, we need funding, we need also a good and strong team. I came myself with a business development background. I worked and I know how it works from the inside of the biotech and pharma industry. Our inventor, our associate, is the Professor Papo. He developed that specific concept, and for this, he won the Pierre Fabre Prize of Therapeutic Innovation. At every single step, we have the right people, and that does include the physicians who tell us every day what the patient needs. It's a very active market. It's a very important market. So join us in that mission, which is to develop, in our mission, to develop the selective chemotherapy safe for patients. Thank you.
Thank you so much, Ori. And that was to the second 5 minutes. Well done. We'll move over to the jury, who I'm sure has some questions for you.
Yes, can I start?
Absolutely.
I would like to ask you a little more about your preclinical proof of concept and data. What have you used so far in terms of models?
We used what we call MIA-PACA2 for pancreas models, and we also use a breast cancer new model. We are now undertaking PDX model to validate it and to get more data on the efficacy and comparison to standard of care. So for scale 1, we are very much at the end of the preclinical non-regulatory stage.
Have you used subcutaneous models so far?
Yes, absolutely.
Who else from the jury wants to add a question?
You said that you have a, well-tolerated, safe technology. So does it mean you completed your toxicology studies already?
We did accurate tox, but not repeated yet. One of the reasons is that if you do look at the drug that we carried, the active agent, which is the auristatin E, a high potent drugs, which cannot be used as a free molecule. It's being used, and that's also well-known drugs because it's been used by a specific ADC. We know that we are able to increase the level of drug carrier, so to transport a lot more drug in a safer manner than the drugs already on the market.
Something else that the jury might be wondering about.
How specific is the enzymatic activity that you need to release your anti-cancerous agent?
Sorry, I didn't get the beginning of the question.
The enzymatic activity. So how specific is this to the tumor microenvironment?
Well, the enzyme, the beta-glucuronidase, is present in all cells of in the body. However, in the tumor microenvironment, it's overexpressed and is expressed extracellularly, which has been already documented for the past 5-10 years. So that's something which is a fact. And we use that specific presence to be a trigger, to be a second level of targeting for the activation of the drug. So that cannot happen anywhere else in the body, outside a solid tumor.
We'll move on to Cem, who also has a question.
Thanks. I have a quick question, actually. About the funding size that you're seeking. Why double-digit and until phase 2 rather than choosing a biteable size? Maybe after phase 1 studies, having another fundraise.
Well, I'm not sure if you get right. Your question is, why do we get? Why are we looking for?
So basically, I look at the sort of, you're looking for a double-digit investment size at the moment.
Okay, I got you.
Okay. Exactly.
Okay, so the reason is because it does include the end of the preclinical, so the regulatory preclinical, which is worth 3 million according to the quotation we have. Plus, if you do 2 development, and that's the purpose of the next 10 million, to do the pancreas and the triple negative at the same time.
Okay.
We have some more time for extra questions by the jury.
Can I ask, can you hear me?
Yes, please, go ahead.
Go ahead.
Sorry, Laura, go ahead. Okay. So I'm not sure I understood the, how do you get this non-toxic effect? Is that something specific with your vectors? I'm not sure I understood your differentiation in terms of the product itself.
Okay, well, the fact is that the standard chemotherapy is active everywhere in the bodies, okay? Whereas here, we are able to active the active agents only at the tumor site. So there's one level which is the accumulation in the tumor microenvironment. So you accumulate, but you're not active. And the second level is thanks to the enzyme, which is overexpressed. Then you accumulate, you release the drugs, which is already accumulated but not active. So with this kind of double targeting, you make that happening only at the tumor and not everywhere else, which is the way that standard chemotherapy works today. Even somehow the antibody drug conjugate are activated sometime a bit outside the target. So double targeting makes a kind of double security to make sure we are activated only at the tumor.
We are.
What is the enzyme?
We have to stop.
Beta-glucuronidase.
I'm sure there's lots more to talk about. And of course, you can connect afterwards as well. The same goes for everyone who is watching. Just go to our matchmaking platform if you want to meet any of the startups or see who other delegates are logged in here today. Yes, thank you so much, Uri. Like I said, right on time, right 5 minutes. Excellent. Seekyo, they are looking at ways to treat cancer patients less invasively. So treat the exact tumor instead of having chemotherapy all throughout the body. And of course, that would mean a major improvement in the quality of life for many cancer patients. We're moving on to our next pitch for this round. Senolytic Therapeutics. They are working to discover novel medicines by targeting cellular senescence. Since 2017, the team is collecting knowledge and expertise in the field to discover a superior generation of senescence-related therapies. Please welcome Tim Cash, who will be presenting today. Good luck, Tim.
Good afternoon, everyone. So everyone can appreciate the difference between the healthy-looking hand on the left and the healthy-looking liver compared to the respective-looking counterparts on the right, and the differences are obvious. What is not so obvious, however, is the macroscopic accumulation of the little brown spots you can see below, known as senescent cells in the unhealthy skin and liver sections. So we are Senolytic Therapeutics. We're located in Barcelona, and what we do is discover and develop early on drugs designed to eliminate senescent cells. So I told you that senescence is associated with human aging and disease, but now basic science has showed senescence causes the processes as well. Here we have 2 old mice that either have or have not had their senescent cells removed by a genetic trick, and also a 3D scan of 2 fibrotic lungs before and after treatment with one of our senolytic drugs, STX-077. You can easily appreciate that the mouse and fibrotic lung without senescent cells looks far better off. First of all, what specifically is a senescent cell? Well, a senescent cell is a once normal cell that undergoes damage and converts into a zombie-like death-resistant state of proliferative arrest, with secreting lots of pro-inflammatory and pro-fibrotic factors. Senescent cells not only accumulate as we age, but they also drive many age-related pathologies, in particular fibrosis, and also the general aging process as well. So our general strategy, as I said, is to both discover and early develop drugs that specifically target and eliminate these senescent cells while not affecting surrounding normal tissues to treat disease and in particular fibrosis. Senolytic Therapeutics was founded on this concept in February 2017 by Dr. Manuel Serrano, a key opinion leader in the field of senescence, who made many of the key discoveries early on in the field, including the existence of senescent cells in vivo. A seed investment was obtained by Life Biosciences in June of 2017, and Senolytic Therapeutics has been running our R&D program focused on developing therapies for fibrosis in various organs, as well as in the field of oncology. In addition to our seed investment, we have been very successful at raising non-dilutive funding in the amount of 3 million euros. And our strategy has been to out-license assets and IP early on during discovery and during the first-in-human stage. We have demonstrated success in this model this year as our oncology platform has recently been out-licensed and incorporated into a newly created company called Rejuversen in Switzerland. The fibrosis program, along with other further discovery pipelines, continues at Senolytic Therapeutics in Barcelona and will also be out-licensed following our next round of investment. So Senolytic Therapeutics and others have now validated that senolytic drugs reduce fibrosis in lung, as you can see the pink fibrosis disappearing with senolytic drugs on the top, in the kidney, as you see the white disappearing, and the liver, as you can see in red on the bottom. So our lead indication for our lead senolytic drug, STX-084, is non-alcoholic hepatosteatosis, or NASH, which fibrosis is the most important clinical predictor. The NASH market is huge, affecting up to 12% of the US population, with the global market expected to reach $37 billion by 2027. The disease itself is characterized by a fatty, damaged, inflamed liver that can lead to life-threatening cirrhosis and liver cancer. The fibrotic stage and clinical outcome in NASH patients is clearly associated with senescent cell burden, as you can appreciate from the graph, and unfortunately, there are no currently approved drugs for NASH. So we will complete the testing of our lead drug, STX-084, in preclinical animal models with existing funding in 2022, and plan to fundraise for 6 million euros more to carry out the preclinical development further along, as well as the phase 1 study in NASH. In addition, we will request an additional 2 million euros to fund the discovery program to identify novel senolytic targets in various age-related pathologies associated with fibrosis, mainly focusing on CRISPR-based screens. So the Senolytic Therapeutics team currently consists of our academic founder, Dr. Manuel Serrano, who, as I said, is a key opinion leader and senior advisor to the company, myself as the CSO, having a background, a long history in academia, as well as in early discovery in biotech, and the acting CEO, Mark Ramos, who has served as CEO of multiple companies, has co-founded multiple companies, and sits on the board and advises multiple companies. Our team also consists of a bench scientist for early discovery work, as well as administrative support. Our value is driven by our ability to identify new targets, validate them through early proof of concept, and develop them to capitalize via out-licensing and new company creation. And our recent success with that was Rejuverescent, as I said, and our focus on developing senolytic drugs for oncology and fibrosis has the potential to extend it to many treatments for different fibrotic diseases associated with COVID, and also to delay the general aging process. So thank you for your attention.
Wrap up there, Tim, because we already are at the 5-minute mark. And that means we're going to listen to our jury and see if there's any questions from them. Who would like to kick off?
I have a question. Thank you, Tim, for an interesting presentation. Do you see any unwanted side effects or damages of the tissue in your current clinical trials, and any toxic effects, anything like that?
Yeah, so we ourselves have not run any clinical trials yet. Our competitors are currently entering that arena in the setting of osteoarthritis and eye diseases, to which they're locally administering senolytic drugs to kind of cut down on those side effects. So as you know, senolytic drugs are a wide category of drugs with a wide category of targets. So it's all going to depend on the target at the end of the day, because the targets that are expressed in senescent cells are most likely expressed in normal tissues. But the hope is that they'll be much more highly expressed in senescent cells compared to normal tissues, so that we can find a therapeutic window there.
Okay. Who from the jury has something else that they would like to have explained?
I would like to ask your talk about senolytic drugs, plural, and as a category. So I wanted to ask you a little bit about the IP in this case. What do you protect here? Yeah,
sure. So our most advanced drug that I described, STX-084, is an in-licensing asset. So that would be IP that's in-licensed. Our goal is to develop our own IP around NCEs that would be derived from the target screening and subsequent drug screening in various platforms and medchem work down the line.
All right, thank you, Tim. Anything else from the jury?
Maybe I have a quick question, actually. In terms of NASH and selecting the candidate, everybody's sort of working on NASH nowadays, and unfortunately, of course, from the payer's perspective, even it's like a lifestyle disease or being even taken as a disease. What is sort of the in terms of the development program and so on? How do you see it? Do you have other alternatives for, again, so on and so forth, for risk management? Unless, I mean, if you see any competitors coming into the marketplace, in a shorter time period.
Right. So usually when we kind of scan these senolytics early on, we don't just focus on one fibrotic condition. We usually scan several. So another favorite of ours is our diseases associated with kidney fibrosis. And And so we've worked with many different models of kidney fibrosis in the last 3 years. We're very familiar with them. So that would kind of be our backup indication.
Thank you.
Okay, someone else from the jury might have a question for Tim. All questions answered?
I have an additional one, which relates to the business model and the out-licensing and formation of new companies once you've validated a target and spun that out. What's the rationale behind this model rather than just having it internally in Senolytics?
I think it's mainly built on the success with our first kind of case study here, where we had an oncology program internally, which somewhat stood out from the fibrosis program. Oncology, of course, can be considered an age-related diseases, but many people like to put it in a separate category. So we were able to find an interested investor to out-license that and not spin it out, but set up a completely different country. So based on that initial success, I guess this is our model moving forward, that we think we can be fairly successful in out-licensing and setting up separate companies early on in discovery moving forward, given the activity in the field of Senolytics.
Okay, thank you.
So we have a little short of 1 minute left for maybe a final question by the jury. Or are all your questions answered right now? I think so. Is there anything else that you would like to add, Tim? You still have a few seconds.
No, that's all from my side. Thank you for your attention.
Okay, thank you very much then, Tim, and thank you, jury, as well. Yes, we're slowly moving towards the last pitch of today, but not before we quickly summarize Senolytic here. They're targeting aging cells for the treatment of, for instance, fibrosis. Important, lots of people are, suffering from this. They already have some investment and funding and are looking to expand their research. It's a large market, and it's only looking now at liver fibrosis, but of course there are many other fields that they can focus on as well. Now, while the jury is entering their scores for this pitch, we go to UVera. And UVera is a startup focusing on skin protection against UV sun radiation. And the team's mission is to provide a natural, safe, strong, and stable, eco-friendly UV protection while minimizing the impact on the environment. Please welcome our final presentation of the day by Adam Kiciak.
Today presenting will be me, Magda Yander.
It's Magda. Okay, I thought that you switched, but it's Magda. Go ahead.
Yeah. Ladies and gentlemen, we've screwed up our planet. Not only while driving, not only while shopping, but also during every single holiday. This is Palau. It's just like another island paradise country. But Palau was the first to look below the waterline. And what they found was terrifying. In 2015, Dr. Craig Downs discovered that oxybenzone, one of the popular UV protector found in the sunscreens, has a toxic effect on the coral reefs, even at the concentration of 1 drop in 6 Olympic-sized swimming pools. Moreover, subsequent studies showed that the popular UV sunscreens are also harmful to human body. This research led Palau to be the whistleblower and to ban certain sunscreens. Hawaii, Mexico, Florida will be imposing the similar restrictions soon. But how come? How it happened that sunscreen destroys our ocean's biodiversity? It turns out that when we wash or when we shower or when we are taking the sun bath, the harmful chemicals which are in the sunscreens, they wash off and they go into other waterways. The scientists were gathering information for the long time about the side effects, but just recently it has been discovered that they destroy sea life. Moreover, they can disrupt hormones, accumulate in the human tissues, and also induce cancer. The good news is that if we act now, we can still put the ocean's biodiversity right. My name is Magdalena Yander, and I'm co-founder of an R&D company. We are producing the next generation healthy and eco-friendly UV protective substance. It's possible thanks to our technology and discovery of specific strain of cyanobacteria. They are producing fully rage UVA, B, and C UV protector. Lab tests shown that it's non-toxic and doesn't penetrate deeper skin layers. Our production It's cost efficient and eco-friendly. We are consuming tons of the carbon dioxide in order to produce oxygen and the fertile biomass as the byproduct. No wonder why cosmetic companies are really interested in our technology. We are targeting multi-billion-dollar sunken market with the product with cosmetic companies which has been searching for years. Our revenue streams are based on 3 models: licensing of our technology to cosmetic companies, selling of the UV protector to the cosmetic manufacturers, as well as the selling of the purified biomass to agriculture and pharma industry. Our products significantly outperform the narrow UV protective substance developed by the startup as the Gadusol and other big cosmetic brands. We are the only one to develop our product from the simulation of the specific microorganism on the industrial scale. Our team consists of the scientists and entrepreneurs who has vast experience in the commercialization of life science projects. Before, I was leading the projects which were licensed to Novartis and Sumitomo in Japan, and I was recognized as the Businesswoman of the Year in the innovation category. Our CMO, Adam Kiciak, has successfully developed 4 medical products and licensed them to pharma industry. Our CFO has closed 3 global licensing deals with the amount exceeding $500 million. We will be able to commercialize our technology by the end of 2023. Next, in 2024, we would like to enter the markets in Asia and subsequently in North America. With 3.5 million euro, we are planning to build pilot biofactory and produce UV protector. We are right now raising 3 million euro to reconstruct and develop our commercial team and also secure regulatory pathway all over the world. We plan to break even in 2023. The way we live on Earth is sending biodiversity into decline. We are the only one to make a difference. Our natural UV protector addresses the strong need of the cosmetic industry and responds to incredible demand for the next generation health product without changing consumer habits. With our solution, you can protect environmental balance and secure the future of our life. Would you like to join us? Thank you.
Thank you so much, Magda. Well done. Yeah, and it's now up to me to, for the final time, ask the jury if they have anything that they would like to know.
Yes, I have a question for Magda. Thank you for the presentation. So could you explain a little more about the product itself? Is this an extract from this bacterial strain or recombinants?
The bacteria is producing the substance outside, so it's not the extract which is taken from the inside bacteria, but it's generating our UV protector outside of it. So we are like taking our UV protector from the whole biomass.
I see.
Anyone else from the jury?
You mentioned that you have 3 different revenue streams potentially. So can you give us a split among those 3?
I may come back to the slide if I can. I see that something was wrong also on the presentation with the finances. Actually, the first revenue stream is from the licensing. We would like to license our technology. So right now we are developing the pilot biofactory, and we will be licensing the whole design and also IP protection for the construction of the bigger scale of that kind of the solution. And we would like also to be a small producers of that kind of the UV protector. So we can also sell the raw material to interested companies, not only from the cosmetic industry, but also from the other applications.
What other explanations might be wanted by the jury?
You do already any dermatological testing?
Yes, we have dermatological testing. Maybe I will put the question to Adam Kiciak, who is our CMO.
Oh, thanks. Yes, certainly, yes. We did some initial testing showing absolutely no toxicity at all. The substance itself is widely known from years, but nobody till that moment could obtain substantial amounts to verify the non-toxic effect on skin as well as on in vitro modeling. So we showed and confirmed the presumed indications that it can be easily and safely used for humans. And what is of note, that the compound itself does not penetrate skin at all.
Thank you, Adam. Are there more questions from Magda?
I have one more question. Have you been able to compare the performance of the bacterial UV protection against sort of what's in drugstores today?
Magdalena?
Yeah, well, the compare, like you mean the UV protection properties, or the
Yes.
well, actually it's exceeding the UV protection properties in case of the coverage of the range UVA, UVB, UVC. It's also having the desirable depletion in the UVB protection in order to maintain the production of the vitamin D by the human body. And at the same time, I would like to focus on the stability of this compound. Like, it's stable for years. So like, we don't have any kind of the discussion about like what is the shelf life of that kind of the product with the cosmetic companies, because we are already in talks with some of the big concerns. So in case of the physical parameters, it's exceeding what right now is on the market. It's the natural product made by the bacteria through the evolution. So we are not playing with the nature.
We have some time left, so should there be anything that you else want to know, go ahead.
For the sea life safety, so what kind of proof do you have that is safe for the sea life?
Adam, will you follow this question?
Yes, please. So we did some measurements showing absolutely no oxygen, I mean the ROEs production, which is of danger, especially for the life ecosystem. And we showed that there is no activity, such activity. Moreover, the compound itself, in case of being in water, will be used as a source of energy for any living animals in the oceans or in the seas. So it's absolutely neutral from the ecosystem perspective.
Time for one quick additional question, if there is one.
There's a couple of competing products on the market, HelioGuard 365 and HelioNoe, based on a little bit different approach, but also using natural ingredients. So what's the main advantage over these products?
Okay, first of all, the great activity. Activity, nothing to compare with the nowadays products. And the only natural comparators we can recognize in the market are oils coming from certain plants. But the problematic issue that they are absolutely not powerful in the UV protection and very weak in stability. They loses its stability within 20 minutes after usage.
And with that, we have to conclude our jury round. Thank you so much, Magda.
Can I add one thing, one sentence?
I'm sorry, we have to be very strict. We all get.
Okay, last year we won EIT Innocence Award, and this year we were granted by the EIC Accelerator with the Green Deal grant. So we have some couple of success in case of the Green Deal issue.
Well, that was our last presenter of the day. Thank you so much, UVera. And thank you all to all the presenters who have done a great job at giving us these amazing pitches. And thank you, jury, for listening to all of them, asking questions and taking notes. Good luck on your deliberation in the jury room.